Hello, we're glad you've joined us for this live webinar, Cold PCR Technology, Current Status and Application in Liquid Biopsies for Cancer and Prenatal Diagnosis. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by PerkinElmer. PerkinElmer is a global leader committed to innovating for a healthier world. Their optimized automated solutions are designed to improve the efficiency of genomic and proteomic workflows. With proprietary nucleic acid isolation technology, flexible liquid handlers, robust library preparation solutions, automated nucleic acid analysis and protein characterization systems, and innovative solutions for single-cell genetic analysis, PerkinElmer is eliminating the challenges associated with genomic and proteomic analysis. To learn more, visit PerkinElmer-appliedgenomics.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Mike G. Makrigiorgos, PhD. Dr. Makrigiorgos is a professor and director of the Medical Physics and Biophysics Division in Radiation Oncology at Dana-Farber Cancer Institute and Brigham and Women's Hospital's Harvard Medical School. He also directs his DNA technology laboratory and a radiation preclinical facility. His research interests include the development of novel DNA technologies for molecular diagnostics in oncology and the detection of circulating cancer biomarkers. Funded continuously by NIH over the last 25 years, he invented several new techniques for molecular diagnostics, including Balanced-PCR, COLD-PCR, and NaME-PrO. He is a member of the Editorial Board of Clinical Chemistry and has published over 150 articles in journals, including Nature Medicine, Nature Biotechnology, Nucleic Acid Research, and Clinical Chemistry. He received his undergraduate degree in physics from the University of Athens, Greece, his PhD in medical physics from the University of Leeds, UK, and his postdoctoral training in radiation biology from Harvard Medical School, Boston. Dr. Makrigiorgos will now begin his presentation.
Yes, good morning, everybody. Thank you very much for the introduction, and I would also like to thank the organizers for inviting me to this webinar to review the history and current applications of cold PCR technology as well as recent developments. So we will talk briefly about the significance of detecting low-level mutations, which is what cold PCR does, and go over the cold PCR principles and early studies, as well as the most advanced version of the technology called Improved and Complete Enrichment, ICE cold PCR, its principles and applications, as well as multiplexed ICE cold PCR. So, mutation detection, as we all know, has numerous applications in cancer diagnostics and personalized medicine. These low-level mutations are frequently, very frequently, a potential problem in clinical samples. From the point of view that always there, they have major clinical significance in many cases, but they are difficult to detect, a lot of technical difficulties. So some examples, low-level mutations are encountered, I would say, in the majority of clinical samples. In cases that one has infiltrating cancers like pancreatic infiltrating adenocarcinoma, in samples obtained from surgical margins, because there you only have traces of mutations, hopefully no mutations at all, but definitely you have a high majority of wild type containing cells, wild type DNA containing cells. In bodily fluids, about which we hear a lot these days, liquid biopsies, circulating tumor cells always have high content of non-cancer cells as well, containing wild-type DNA. In non-microdissected samples, in colonoscopies, where you want to detect things like microsatellite instability very early in the process, you have an excess of normal DNA as well, and in cases of genetic mosaicism. But even more so, even in samples where you have the majority of the cells being cancer cells, even then you still have intratumoral heterogeneity, as this landmark paper demonstrated back in 2012. It is from the group of Charles Swanton in England. They sequenced different parts of the tumor, and it was shown that different mutations belong to different sections of the tumor, as well as some common mutations, the truncal mutations that are present in every cell. So a lot of intratumoral heterogeneity, and that obviously brings up both technical and conceptual problems as to what is a biopsy. How much of the tumor molecular profile does a biopsy capture? Now, thankfully, things like liquid biopsy have also grown recently and demonstrated that there is circulating DNA from the tumor in the blood, which in fact may represent several parts of the tumor, several subclones of the tumor, as well as hidden tumor. And therefore, this can provide complementary information on the molecular profile of the tumor, since many different subclones may be represented in a blood draw from which one isolates circulating DNA and tumor profile. Now, in addition to applications of low-level mutations and liquid biopsy in the field of cancer, I would like to mention that also in cases like in medical applications like prenatal diagnosis, infectious diseases, and in detection of transplant rejection, which are all done via detecting circulating DNA in blood. Again, we have the same issue of few mutated DNA molecules in an excess of wild-type molecules. So as you can see, the applications of low-level mutation detection are quite general, and they apply not only to cancer, but also to other fields that are emerging or established. So there are several challenges, however, technical challenges in measuring circulating DNA, and some of these are shown here. As we said, you have rare mutant copies in a wild-type background. DNA is highly fragmented in blood. It's like the majority is a result of apoptosis in tumor cells as well as normal cells. So that makes it highly fragmented and low molecular weight, typically 150 base pairs DNA or less. But the difficulties continue. You have a limited number of DNA molecules, nanograms per ml of plasma or blood. So as a result, from a standard collection of 10 to 20 milliliters of blood, you only get a few nanograms of circulating DNA out of which to extract the small portion that is useful information, the small portion that comes from a tumor or from a fetus in the case of prenatal diagnosis. Just have a few precious nanograms to work with. And as we said earlier, also tumors are genetically heterogeneous, so different clones can shed different amounts of circulating DNA characterizing the molecular profile of the clone, so that compounds the difficulties. So technologies that have been shown over the last 10 years to do a good job in characterizing circulating DNA and measuring the molecular profile of the tumor are NGS and digital PCR, among others. These are two established technologies, and I have listed the basic characteristics of each one of these two technologies below that. As we all know, NGS is highly parallel. It can be a discovery tool because you can have a very big net to catch fish in there. Basically, you can screen thousands of gene targets at the same time to discover new mutations depending on what therapy is applied and so on. It can also be a diagnostic tool when it is used with focused panels, and it's also quantitative, which is in some applications it's an important factor. On the other hand, whatever we do is still remains expensive, even if the price is reduced as we expect. It's still not going to be a cheap technology like real-time PCR, for example, or digital PCR, and it still takes time to analyze the results, and you need an instrument or a core facility to perform that for you. On the other hand, digital PCR, it is wonderful from the point of view of absolute quantification. It is very reliable, but it can only do one or a few targets at a time. And again, you need an instrument. So today I'm going to talk about COLD-PCR, which is an even more flexible technology to use and to adapt to various clinical scenarios, and which can be applied on a standard real-time PCR or standard PCR instrument, which is available in every lab. So our approach through the years has been to apply enrichment of mutations prior to their detection. As you can see here, you have many of wild-type molecules and few mutant molecules entering the funnel, and then by applying COLD-PCR, one can enrich the mutant, and then the endpoint detection method is becoming much easier to perform. So COLD-PCR has been around now for 10 years, so it's a good anniversary, a 10th year anniversary of the technology. And in a big view, a helicopter view of what has happened these 10 years, is that several papers have been published. I'm only listing here, I would say, the main papers that have driven the technology. although it has been translated to many different applications by different groups. There is more than 1,000 references in the literature in COLD-PCR. If you check Medline, and there is at least 50 different groups around the world that have used the technology for their own needs and adapted it. And in terms of commercial application, the technology has been licensed to Transgenomic, Precipio, companies that have merged now. For the most advanced version of the technology, which is the LNA-based iSCOLD-PCR or ICP, about which we'll talk tomorrow, later on today. So here is the principle of COLD-PCR, as it was originally published. And it's important to understand that in order to understand the later versions of the technology. So upon denaturation at 94 degrees, the two strands of DNA are separated, and then instead of going all the way down to a temperature to which the primers will bind, we stop at an intermediate temperature, like 72, 75 degrees, 70 degrees, that allows the top and bottom strands of DNA to bind with each other without yet the primers initiating synthesis, because we keep the temperature high enough so that the primers don't bind. So obviously, if you use a hybridization at 70 degrees, the primer should not appreciably bind at 70 degrees in order for this to work. At the hybridization temperature of, say, 70 degrees, the top and bottom strands will bind, and if you have mutant, that is more likely to hybridize with wild-type DNA because this is in excess, and then you create a small bubble. And this bubble contains mutant on one side and wild type on the bottom strand. And you also create a lot of wild type homoduplexes. Then by increasing the temperature by a few degrees to a critical temperature that differentiates between homoduplexes and heteroduplexes, you achieve preferential denaturation of the heteroduplexes. So here you denature mutant and wild type strands. The homoduplexes remain hybridized. And following that, then we reduce the temperature to, say, binding temperature of 60 degrees. The primers bind preferentially to the denatured homoduplex that contains the mutant, and we synthesize preferentially the mutant sequences. And then we continue this process 20, 30 cycles, and the enrichment is gradually compounded. And the result is that at the end, the net effect, the generated PCR product contains a much higher mutant sequence as opposed to wild type relative to what we started. What is important to note is that mutation enrichment occurs at all positions on the sequence. It doesn't matter where the bubble will form. You will always have some enrichment, which can be different from position to position, but in all cases you get an advantage and you increase the sensitivity of mutation detection. This makes COLD-PCR distinct to other technologies that utilize changes in the primer sequence. Like at the end of the primer, you can have allele-specific PCR that enriches for mutations that are right on the 3' end. And that is another way to enrich mutations, which, however, work only for one position at the end of the primer. Here you can have 150, 200 base pairs of amplicon, and irrespective where the mutation is, it will be enriched. So as a result, COLD-PCR can be combined with technologies, endpoint technologies for mutation detection that detect mutations anywhere on the sequence, and that's the only technology that can do that, like sequencing, for example, or high-resolution melting. So as you see here, since PCR is performed prior to almost all methods for mutation detection, like HRM, dideoxy sequencing, pyrosequencing, NGS, in many cases you perform some PCR step, RFLP, and so on. If you replace the initial step with COLD-PCR, you increase the sensitivity of all these methods simultaneously. So let's look at some examples. Improvement of sequencing sensitivity. So this is some results from the original publication where we have a mutation in p53, and on the left side you see if you perform regular PCR followed by Sanger sequence, the mutation is hardly visible. If you perform COLD-PCR with denaturation at critical temperature 86.5, the mutation is enriched and it can be seen. And the same thing from plasma circulating DNA in the middle section. Regular PCR on the left, you can't see the mutation, but you can see it becomes heterozygous essentially if you perform COLD-PCR. And the same thing at the bottom, using clinical FFP specimens from lung cancer patients. You can barely see the mutation with regular PCR, but you can see it. It's very evident with COLD-PCR. So irrespective of where the mutation is, it will be enriched. Now, you may ask the obvious thing, what if you do one round of COLD-PCR and then you do a second round of COLD-PCR, a nested step for COLD-PCR? Will the enrichment increase even more? And the answer is yes, as you see from that paper back in Human Mutation back in 2009. With regular PCR, you don't see the mutation. That was a very low level mutation that we applied here. Even one round of COLD-PCR here does not show the mutation. 2 rounds will eventually show the mutation without changing the profile of all the other sequences. And that's important to state that during COLD-PCR, since you are doing continuous cycling, you may have a polymerase introduced error. So if an error is introduced, say at cycle 5, then this false mutation will also be enriched during COLD-PCR. So eventually, if you keep doing too much COLD-PCR, you will produce false positives because of polymerase misincorporation. Here we demonstrated by having a proofreading enzyme that up to 2 rounds of COLD-PCR you don't see appreciable generation of mutants, at least at the levels that we can see by Sanger sequencing. So bottom line, 2 rounds of COLD-PCR increase the enrichment even more. And an application now of COLD-PCR in conjunction with sequencing. In this case, what we did is we had 2 rounds of cold PCR, and as a control experiment on the left, we had 2 rounds of conventional PCR with exactly the same primers. And then we went ahead and sequenced using the HiSeq 2000, and that was back in 2011 that we performed this study. And what we saw here is essentially the limits of sequencing. By using conventional PCR, a 2% mutation can still be seen over the sequencing noise that we get by using the standard protocols that comes from Illumina, while with 1% we are close to the noise. Okay, so basically that's the limits of standard sequencing, unless one applies the newest development using molecular barcodes. But I'm talking now about the standard application that comes with the commonly used protocols. If, on the other hand, one applies cold PCR before going to the standard Illumina sequencing, that's what you get. You increase the signal while keeping the background approximately the same, and that you can see that you can go all the way down to 0.02% of cold PCR and still discriminate over the background. Okay, so by enriching the signal, you increase the signal to noise ratio, so you can detect via sequencing the lower level mutations. And an important point to notice here is that you just need a small sequencing depth. You can see here that in the extreme case we detect a 0.02% mutation with just 28 reads, sequencing reads, which is impossible unless one does some enrichment of the mutants. By enriching the mutants, you bring them to a high enough level that 28 reads are enough to show the signal. So 2 advantages: increasing the signal to noise ratio and a reduction in the number of sequencing reads that you need. So another application is the improvement in real-time PCR, and we use TaqMan here and perform cold PCR. What we have is 2 TaqMan probes, one that matches the mutant and one that matches the wild type sequence, and we perform the TaqMan real-time PCR under cold PCR conditions. The cycling conditions are, as I explained earlier, in the cold PCR principle. And here is a comparison. On the top panel, you see a conventional TaqMan genotyping of the T790 EGFR resistance mutation. And under this approach with conventional TaqMan, you can just differentiate about 12% mutant to wild type from the signal that you get from pure wild type. You cannot go any lower. If you apply the very same primers, the very same probes using cold PCR cycling, then you can go down to 0.8% mutant to wild type. And this actually comes to be a very quantitative test. So if you are talking about applying cold PCR on the same mutation every time, in real time, you can get a straight line like you get for regular PCR versus concentration of the original sample and mutation abundance of the original sample. I have some examples on that later. Next is the improvement of high-resolution melting by using cold PCR prior to melting. Now, high-resolution melting, as you know, is one of the most commonly used methods for mutation detection because of the reliability and the ease of use and the very low cost. These days, a lot of PCR instruments come with a standard high-resolution melting mode. So, obviously, application with COLD-PCR is something we wanted to consider here, especially since COLD-PCR enriches mutations anywhere on the sequence, and HRM detects mutations anywhere on the sequence by using mutation scanning HRM. So the two technologies work together very well. So what you see here is at the top a serial dilution of samples that have 10% mutant, 8% mutant, 6% mutant, and so on, serial dilution with conventional HRM, conventional PCR followed by standard HRM, and you can see that the limit of detection is 3 to 5% here, depending also on what mutations you have. So that's the standard technology. If you apply COLD-PCR, you get a very significant improvement in the LOD, the lower limit of detection. 0.1 to 0.5% can be detected very easily. And importantly, one is able to sequence the product because although HRM is very easy to use and basically very low cost and low effort, it won't tell you where the mutation is. And if you try to sequence it, very often you can't find. For example, in the conventional HRM, a 5% mutation may be detected by HRM, but then you can't sequence it by using Sanger. By doing enrichment, you can both detect lower level and sequence them. The enrichment is big enough that you can see it on Sanger. So that was also early work. COLD-PCR HRM overall is the easiest way to exploit the technology. Immediately after COLD-PCR, you perform the HRM without necessarily having to open the tube. There is no new instruments or reagents needed. In most cases, there is no need to change primers, and it enables increasing sensitivity and sequencing. So I would recommend to anybody who wants to start using COLD-PCR to combine it first with HRM to make sure everything works okay, even as a quality control test. So I compiled here a number of reports on COLD-PCR over the last 10 years that have been applied in different fields for detection of mutations in tumor margins, treatment assessment in colorectal cancer, circulating tumor cells, but beyond cancer, prenatal diagnosis, crop improvement, drug-resistant mutations, mosaic mutations, and so on, as well as modifications of COLD-PCR technology to be applied in DNA damage detection as opposed to mutation detection. So most of these have used HRM, the majority, not all, but HRM again is the easiest way to exploit the technology, followed by sequencing if you see a positive on HRM. So now let's move to the more advanced part of the technology, COLD-PCR. Improved and complete enrichment. We developed that in 2011. And why we developed it? We noticed that in the early cycles of PCR, it is more difficult to form a heteroduplex because you have very low levels of your template. So in the first 10 cycles, when you start from genomic DNA, you have very, very few molecules. So they can find each other in solution. The heteroduplex doesn't form. So for this reason, we thought, let's help the situation and add a reference sequence, which is essentially just a wild type sequence, which by itself cannot be amplified by the primers. You don't want to amplify the reference sequence. What you wanted to do is that in early PCR cycles, since it has a high concentration, it will offer wild type template to bind with your few mutant molecules in early PCR cycles. So by adding an excess of reference sequence in the solution, we help hybridization and heteroduplex formation at early PCR cycles. And then the process produces an improved and complete enrichment, and that's why we called it ICE COLD-PCR. Now, the other thing, of course, is that since you are including a synthetic reference sequence, you can synthesize it either from an oligonucleotide synthesizer or via PCR, like we did in the original paper. You can always include modified bases in there. And in some cases, modified bases like lock nucleic acid or PNA, peptide nucleic acid, or other similar modifications can give you an advantage because they create a bigger difference between heteroduplexes and homoduplexes. To make a long story short, that led to the LNA-based ICE COLD-PCR, which we often, for convenience, we call it ICP. That's the most advanced version of the technology and produces the best enrichment. And I have here some data that was presented by Richardson and co-workers at ASCO back in 2011. That is the first report on the LNA-based ICE COLD-PCR or ICP. You can see detection of BRAF mutations in melanoma. And here in a serial dilution study, you can see that the detection is easily down to 0.02005% mutant to wild type. And here is the same application for EGFR exon 19 deletions. For deletions, the technology works particularly well because, as you can imagine, a deletion or an insertion makes an even bigger heteroduplex difference than a single point mutation. And one can sequence through the deletion after you enrich it to the extent that COLD-PCR does. So it's, you can see here a very nice sequencing through the deletion. Yes, that was presented in 2012. And then in 2013, the group of Jörg Tost in France also independently developed an LNA-based ICE COLD-PCR, which they called e-ICE COLD-PCR. It's essentially the same or very similar to what I described. They have done an excellent work working out a number of advantages of this technology. And I have some slides that were kindly provided by Jörg Tost to show you in some of the next slides. So here you see the comparison on the left of conventional PCR. We have 3 cancer samples and 1 wild type. And in the middle we have COLD-PCR. And on the right, we have ICE COLD-PCR. And as you can see, there are cases that conventional PCR and the originally developed cold-PCR don't solve the mutation. It is a very low-level mutation, but it becomes clearly evident with ICP or ice cold PCR. Yeah. At the same time, the wild type remains wild type. That's always important to verify in such sensitive technologies. So one question that many people ask, and we also asked through the years, is how do you quantify the original mutation abundance when you create this enrichment of cold PCR, which can be different from mutation to mutation and from point to point in an amplicon, which is 150 base pair long? How do you find the original mutation abundance? This is not always necessary, but in some cases, some applications like monitoring for minimal residual disease can be important so that you know how the tumor dynamics are developing. So here is a serial dilution for 5 different mutations using ice cold PCR, and what we have plotted here on the x-axis is the amount of alteration in the starting sample, right? So this is what you want to find out. What is the amount, the mutant allelic frequency in the starting sample, and on the y-axis is the resulting frequency after cold PCR. And as you can see here, if you take any given mutation, just concentrate on the green color, for example, you can plot a nice, relatively good straight line for the same mutation on the same amplicon. But for different mutations, the enrichment is different, so the relation is different. So it is not possible to have a single relation that will tell you what the enrichment is, irrespective of where on the sequence it has taken place. But for a given mutation, like for example a T790, you can very easily relate the obtained final mutation enrichment to the original alteration. So it's quantitative only if you consider one mutation at a time. And again, this is a slide that we received by York Tost Group in France, who has done detailed such studies. They have compared that to digital PCR. So they took individual mutations and they compared to quantification that they get with ice cold PCR as compared to digital PCR. And as you see at the bottom in the 3 graphs that are presented here, you have nice straight lines in all cases. As long as the enrichment you obtain is calibrated via a serial dilution curve, it doesn't change anymore, and one can apply that to clinical samples for a given mutation at a time, and you get results that are not very different from digital PCR. Of course, digital PCR is an absolute quantification method. Ice cold PCR is a relative quantification method. You need to create the standard curve before you reach something like this. Okay. So, and here is a combination of the ICP, the LNA-based PCR, and TACMAN at the same time. Again, showing essentially the same thing. On one hand, that you can detect down to 0.01% mutation, depending also on which mutation, of course. But at the same time that you get a reasonably good straight line, considering the enrichment effect. So the take-home message is that if you restrict it to a given mutation, you can apply quantitative ice-cold-PCR. And this is what we did in this study. So in this study, we asked the question, what level of mutations is clinically significant in the particular case of myelodysplastic syndrome? So that's a collaboration we did with Ben Ebert and Raphael Bezar here at Dana-Farber and with Catherine Richardson at Transgenomic. We wanted to see if low-level NRAS mutations can predict who will recur and who will progress to leukemia, which is a common development of myelodysplastic syndromes. So Ben Ebert and Raphael in their studies have shown that NRAS mutations are prognostic, but we didn't know what about a very low level of NRAS mutation. What level of mutation should we be worried at? So we applied a study using ice-cold PCR to detect the very low-level mutations. Here are the results. We screened 295 samples using either standard cold PCR sequencing or ice-cold PCR, followed by sequencing, and the conventional PCR using MALDI or Sanger detected NRAS mutations. And as anticipated, ice-cold PCR followed by NGS using ion torrent in this case detected many more mutations at very low frequency. Then we asked the question, are these clinically significant? And quantification, as I mentioned before, can be done for individual mutations by doing a serial dilution study and setting up essentially calibration curves. Here is what you see for 3 different NRAS mutations. That's what we use for quantification, and we drew our calibration curves. And here is the comparison with the clinical results, the Kaplan-Meier curves. And what we demonstrated is that if the ice-cold PCR detected mutations are of the order of 0.1 to 0.2%, there is no difference. They have no relevance for development to leukemia, to AML, basically. So they are not clinically significant. However, if they are more than 2% or 0.5%, which was shown to be the threshold, there is clinical significance, and it was shown in this paper that they confer an independent prognostic factor. So here is a case that one needs to detect the mutations quantitatively. You can do it with ICP, or potentially you can also do it with digital PCR, okay, if you have the instrument. And the level of significance is 0.5% in this case. So the level of significance obviously depends on the conditions you are studying. So it will be different for every different study. So further applications of the ICP technology with liquid biopsies are listed here for anyone that has time to read that, basically. But many nice studies have been shown with recently, and they keep increasing for application of ice-cold PCR with liquid biopsies in several clinical situations. The 3 first papers at the top are for cancer. The 2 papers below that are for prenatal diagnosis. Now, for prenatal diagnosis in particular, what one is interested is to detect the paternally inherited mutations in maternal blood. That's what the technology is good for, because these are going to be low-level mutations, and you need to enrich them and detect them with confidence. So the first paper was basically published by Galbiati and a co-workers group in Italy in 2011. And the most recent papers, there have been some intermediate papers, but the most recent papers were published 2018 by my collaborators in Cyprus, where they were applied to detecting paternally inherited mutations in beta thalassemia. So the last few minutes that are remaining, I would like to mention the latest developments of the technology multiplexing ice-cold PCR. Multiplexing ice-cold PCR was first reported by the group of Jörg Tost in France, and where they demonstrated multiplexing of three different amplicons. Here, BRAF, KRAS, and NRAS, and the technology retains the sensitivity for all three amplicons at a time. So you have three reference sequences working at the same time, and with a single thermocycling protocol. So in collaboration with Precipio, we have also been working and developing such systems, and what you see here is multiplexing of four amplicons for EGFR, exons 18 to 21. What we have is sample collection, DNA isolation, multiplex ice-cold PCR, and at that point we demonstrated that a high-resolution melting screening can be very beneficial to tell you if there is a mutation in any of the four amplicons. If there is not, you can stop. HRM is indeed very reliable. So it will save you the work and the expense going to endpoint detection. So here is an example. We demonstrated that a number of different KRAS mutations can be differentiated down to very low levels, 0.1, 0.05% from wild type with HRM prior to doing any other endpoint detection. So you don't know where the mutation or which type of the mutation is there, but you know there is a mutation. And by performing HRM after ice-cold PCR, all in a single tube, you don't need to open the tube necessarily. You already know if there is a mutation anywhere on this sequence. And the same thing for the other sequences at the same time. And now, if it is shown by HRM that there is a mutation, how do you find the mutation? You can either perform sequencing or, like we demonstrate here, you can perform a second PCR, a nested PCR, for each individual mutations by applying a standard Tuckman real-time PCR. So that now will tell you exactly which mutation you have. So you have a series of reactions, each one testing for interrogating one particular mutation. And that is a very reliable application. And here we basically demonstrated that Sanger sequencing also works after multiplexing the four EGFR amplicons, exon 18, exon 19. One can go down to very low levels. Standard PCR on the left, ice-cold PCR on the right. You don't see the mutation on the left, you see it on the right. And that's exon 20 and exon 21. So these data were provided by me, by Ben Legendre and Grant Wu at Precipio, who are our collaborators developing these assays. So in summary, the COLD-PCR technology is very versatile. It enables sensitive sequencing for personalized medicine. You don't need an instrument. You get a high sensitivity at very little cost, essentially. It enables higher mutation sensitivity for every downstream technology you choose, whether it is NGS or real-time PCR, pyro sequencing, or other. No specialized instrumentation, rapid sample to result, and multiplexing capability, which is the emerging application of the order of 2 to 10 targets. That's where we see the usefulness. If you go to even more targets, basically you can potentially choose NGS. If you want one target, you can potentially do ddPCR if you have the instrument. But for targets that you want to get to the answer rapidly without much expense, potentially before you do NGS, we see that ice-cold PCR is a good solution. And because of the flexibility, we can see also application of cold PCR in microfluidic all-in-one devices that are now coming basically to common use. For very fast PCR, within minutes, you can get the sample to result. So in the bottom here, I have listed all the various specimens that one can examine. You have sputum, other bodily fluids that could be urine, of course, and liquid biopsies. And I would like to thank you for your attention and thank my collaborators as well as the people at the lab over the last several years that did this work. Thank you for your attention.
Thank you, Dr. Makrigiorgos, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dr. Makrigiorgos will answer as many questions as time permits. The first question is, how can you determine the level of enrichment due to cold PCR so your qPCR result actually tells you what frequency the mutation is?
Yes, so I hope that some of my slides that perhaps were presented after the question was posed answer this one. Essentially, how do you quantify the original mutation abundance? And again, if you have a particular mutation in mind that you are interested to quantify, you have to run a serial dilution to create a calibration curve. You only do that once for each amplicon and for each mutation. You only need to do that once. It is some work, but that's how you calibrate your final mutation enrichment based on the original mutation abundance. And this is what you do if you want to do it with qPCR. Essentially, that's what you do. You need to do. So it's a similar process. The only technology that you avoid doing that is digital PCR, because that's an absolute quantification. But in most other situations, including cold PCR, each mutation needs its own standard curve, let's say.
Thank you. How does one identify the critical denaturation temperature used in the cold PCR thermal cycling protocol?
Yeah, so this is the most commonly asked question, basically, about cold PCR. So you need to run a gradient for every amplicon that you're going to work with. You need to run a gradient of reactions at different denaturation temperatures and choose the correct one. If you are running ice-cold PCR in the presence of LNA-containing reference sequence, you have a big range to play with. Like as long as you are within 3 to 4 degrees from the best critical denaturation temperature, you get good enrichment, basically. Now, if you are using cold PCR as it was originally described without the reference sequence, there you need to be much more precise. You need it better than 0.5 degrees. So you need a very fine temperature gradient with different denaturation temperatures, and out of those you need to select those that lead to best enrichment as detected with, say, Sanger sequencing or another technology, digital PCR for better accuracy. Now, that may seem as a lot of work, but you only do that once. And for every mutation along the sequence, you always get good enrichment at a single denaturation temperature. You don't need to do that for every mutation. You get good enrichment. You can find good and critical temperature that provides good enrichment along the entire sequence by doing this gradient approach.
Thank you. Next one is, are you performing high-resolution melting right after multiplex PCR on all amplicons at the same time?
Yes, this is correct. Like in multiplex PCR approaches, where all amplicons enrich mutations at the same time, you have essentially a new way to apply HRM, because HRM so far has been applied on one amplicon at a time. But if you have a technology that provides enrichment on all of them at the same time, you can perform HRM on all, say, 4 amplicons or 10 amplicons at the same time. I would say the limit is about 5 to 10 amplicons in this approach. HRM becomes a very strong tool for a few amplicons that are enriched all at the same time.
Thank you. It looks like we have time for one more question. How many amplicons max can you include simultaneously in the multiplexed ice-cold PCR protocol?
Essentially as many as you can optimize. There is no limit. Now we're developing approaches that can go to 30, 40, 50 amplicons. But of course, if you are going to go to so many mutations, so many amplicons, your endpoint should probably be NGS. If you're going to do it fast and rapid and easily at low expense by using technologies like HRM, I would say the maximum is 5 to 10 mutations. So the limitation comes from the HRM side here, not from the cold PCR side.
I would like to once again thank Dr. Makrigiorgos for his presentation. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, PerkinElmer, for underwriting today's educational webcast. This webcast can be viewed on demand through May 2019. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.